All right. Hello once again, and welcome to this edition of the RotaChrome webinar series. Thank you for joining us today. I see that we are quite well um, visited, so let's just start it. My name is András Gáspár, and I'm the Chief Product Officer here at RotaChrome Technologies. I will be your host for today, and as we take a behind-the-scene look at the working CPC system of ours. Before we jump into the tour, here are a little bit of a background about RotaChrome and our technology. RotaChrome is a technology company on the front lines of purification solutions for a myriad of industries. We manufacture world-class chromatographic platforms widely used for botanical extract purification within the pharmaceutical, nutraceutical, and botanical industries. Notable, we created the first industrial-scale CPC or centrifugal partition chromatograph solution designed for large-scale applications. The platform allows companies to expand high purity production capacity with flexibility and scalability while decreasing environmental impacts. A few housekeeping items. Shortly, we will be giving you a tour of our working system. Please note that during the tour and after, your chat feature will remain disabled along with your camera and microphones. Following the virtual tour, we will have some time for questions, so please submit your question via the Q&A feature, which should be a button at the bottom of your Zoom screens. So you can also email your questions to sales@rotachrome.com. If you do not get to your question, don't worry. Our team will get back to you individually following the webinar. I'm very excited to be able to show you all how our technology platform works. So let's just jump into the tour.
Welcome to this virtual tour of RotaChrom's CPC technology. This is the typical configuration of the RCPC. You can see the main unit in front of us, with the control screen on top of it. The configuration will depend on how the layout in that building will be set up. Behind it, in that little table, you see the control unit, which controls the whole flow of the operations. As you can see on the top of the shelf, we've got the upper phase and the lower phase, which will be used for the separation. On the top of the control unit, in this smaller container, is the put material being processed. As you've seen on the previous webinars, the operation of the lower phase has to be fixed and then settled, and then put into these two containers. You leave them on the top of the shelf in order to minimize the strain on the pump. These units in front of me are controlling the whole operation. This selector on the black selector, it decides on the direction of the operations. This machine, the small machine, can burn both ascendant and descendant mode. That's important depending on the fractions that people, our customers, are collecting, and what are the compounds that are important for them. In the typical operation of a THC removal, CBD comes first and THC comes last. If somebody's looking for the THC isolation, then they potentially change that process to optimize the flow. In terms of setting the space up, it is a standard lab environment where we need to have the temperature that keeps the operations consistent. People would underestimate the importance of temperature variation. If the room starts at 68 Fahrenheit in the morning and finishes by 78 in the evening, the chromatograms will shift. We recommend that there's standard air conditioning in this room. Certainly, as a lab safety, we would need to have the proper safety precautions. What happens if somebody accidentally spills solvent? In that case, we need to have an emergency zone. It's very important that the unit is grounded. There is the green-yellow cable on the side, which has to be on a separate grounding. This is a standard configuration. The machine can collect 9 fractions. In this particular case, the customer decided to have 3 groups of fractions. The first would be phase, but then from 2 till 3 or 4, they can collect the CBD without THC. Then they can have foam, which is a combined material. Then at the end of the process, they would collect the THC, which they need to dispose of according to the local regulations. This is the control unit for the PCPC system. The PCPC system consists of a number of elements, including the ICPC. On this controlled unit on the left side, you can see the organic solvent 1, 2, water, and recycle solvent. Those are the materials that come into the system and get into the mixer. The mixer currently shows that it is at 32 Celsius and it's 26% full. The mixer controls the input materials, and the settler will determine whether the upper and lower phase buffer containers will be filled. As long as the temperatures are appropriate, the settler would send back the material to the mixer. Those liquid containers have heat jackets, which will show inside to have a consistent temperature. As we talked about in the small machine, the room temperature impacts the chromatogram and the consistency of the separation. Those units have a set temperature, usually at 89 Fahrenheit. On this control screen, we also see the same home container, which is feeding the ICPC. The 9 fractions that are collected out on the top right. In this case, the system was reset to 0. That's why you don't see different estimated levels. Otherwise, sensors would show how much material is in there. On this particular screen, we also control the impurities processing. From the ICPC, the material would be compounds of materials going to the 9 fractions. The impurities going to the settler in the bottom of your screen. The settler decides whether it's upper or lower phase and sends it to the corresponding tube evaporators. If you mind going to the room, we can identify these elements inside. It's important to mention that this control cabinet is a non-hazardous zone. In the building, when someone builds this space, you need to designate the hazardous and non-hazardous environment. This is the sample circulation switch that the customer will choose to circulate their sample, keeping the rate constant. In this environment, that uses hexane and methanol, which are heavier than air. We need to have it only at sensors that would indicate if anything hazardous enters the environment. The left side will act as a gas sensor, and on the right side, an emergency dock, which is about 10 inches above the ground, so in case a spillage happens, then it can immediately suck out the air from this room to minimize any risk. The smaller container is the sample tank, which includes the material to be purified. It has a heat jacket to keep the temperature the same as the input buffer skin temperatures. In order to avoid any problem with the consistency changes of the input material, there is a circulation which has been implemented that is making sure that the materials can be circulated while it is being processed by the machine. This is the retrofit. They don't have that in other places. As we enter the room, customers have different choices where they want to keep the fresh solvents and the recycled solvents that feed the system. In this particular place, the customer decided to have these drums within the room. In many cases, these drums will be on the other side of the wall. In some cases, customers even place doors to outside in temperature-controlled contemporary, especially if they're using 1,000-liter totes. These input drums are connected by piping to the mixer on the right, which mixes the solvents, as we saw on the control screen. The settler will then feed to the 2 containers in the back. All these containers, the large containers, are 66 gallons in capacity and have a temperature jacket which keeps the temperature constant. The smaller tank in front of the 2 buffer containers is the sample tank or input tank. It's a 15-gallon container. This is where the input material is fed into the system. As you can see, all of these 2 units are coming up from a steel platform. This is part of a scale set up for easier transportation. At this point, I'd like to highlight the height of the room. The room is about 12 feet tall. The recommended height is 14 feet. The main reason for that, and on the top of the buffer tanks, you can see the blue cap level sensors. Those machines, those sensors, need to be taken out for servicing, and you would have to double the height in this room, so you don't need to uproot the scale and take it out of the room. In this particular case, the customer was taking the approach that they would be opening the ceiling if necessary and servicing it through that. I would also like to point out the big table that has the piping, that is nitrogen. All these containers have a nitrogen blanket on top of the liquid. Instead of oxygen or air, which would be a higher risk, a nitrogen blanket is covering the liquid. As we are looking back from the system on the cable rack, which is also installed by the RotaChrom's service technicians, the machine, the ICPC, is fed, and that's part of our buildup. Also, we ask our customers and recommend that they install filters throughout the processing line. In this case, the filter in front of us, we have the material that has the compounds of interest, and it is filtered and then moves over to the other room where the COI evaporation is performed. The pumps that we use in this arrangement are air pumps in order to minimize any risk and have come to use a reliable version. RotaChrome asked the customer to provide an empty room with the appropriate connections for the auxiliary services. When it comes to cabling, RotaChrome technicians will build the cable trays and the cables and the wall penetration, which is a fireproof wall penetration, meeting out to the control unit. The one limitation we have in placement of the control unit is the distance from the ICPC. It has to be less than 30 feet because there is an optical cable which feeds the UV light. The UV sensors result in the control cabinet. This arrangement certainly is much closer to the ICPC. That's an optimal arrangement. The 9 fraction collectors, some customers choose to roll this out and process it in another room. That's why it is on rollers. As you see, there is the nitrogen blanket collection on top of these. This particular arrangement enables draining the tanks by these valves up here by increasing the nitrogen pressure in the tanks and just pushing out the material from these 2 different vessels that they want to use to move the material. This is the part of the system, the ICPC and the rotor. The column equivalent is 33 feet. As you can see, there are 3 wheels behind each other, each with 25 cells. They are interconnected, forming that 33 feet tall column. This is controlled by the control cabinet outside and is able to separate 9 fractions. The machine, because it's larger in size than the RCPC, is running at a lower revolution per minute, usually around 700 to 800 revolutions per minute, while the ICPC runs closer to 900 to 1000. Here on the side, you can see the valves, which are switching to different fractions automatically, according to the program. Those feed into these 9 fraction collector tanks. The volume of these tanks is 10 gallons each, so in total it would be about 90 gallons. If you look at this room, this will have about 280 to 300 gallons of capacity, depending on the arrangement with the first solvent containers that we've seen on the left side of the room. Many fire marshals would limit the maximum allowed capacity to what we see in this room. That's why we are going to the other side of the wall, where we have the solvent recovery solution. In some jurisdictions, we saw the fire marshals will allow all of this equipment in the same room. They require only a C1B2 environment. Customers often choose to install C1B2 equipment. That mostly is visible when it comes to electricity, light fixtures, and cameras. The C1B1 equipment is significantly more expensive than the C1B2 equipment. As you can see, a rotor leg that was manually moved will stop. It does not bounce back and forth. That means that it's stool. It's completely balanced. We have problems when customers decide to move this equipment and they don't take precautions to properly prepare it for transportation. That's when those imbalances can cause damage, which requires service. Of course, during operation, the door is closed. The room is closed, and no operator needs to be in this room. The machine is controlled by the control cabinet outside, operating without human supervision. There's another gas sensor in this room, in case it's required. This is a closed-loop system. No solvent is coming out from the system while it's operating. This control cabinet is the control for the COI, the Compound of Interest Solvent Recovery Solution. It has 2 buffer tanks and 2 evaporators. This is an add-on system which customers can purchase in addition to the CPC. RotaChrome describes in the site requirement the auxiliary servicing the system needs. This example is the customer solution for providing the hot water and the chilling water and the temporary water for the system. There can be other arrangements, but this is how they choose to solve that requirement. We are in the solvent recovery room. This has 2 skids. The one in the back is for recovering the solvents from the base streams. It has 2 containers. One is for the upper phase, the other is for the lower phase impurities, and then the corresponding evaporators. The main purpose in that back end is to recover the solvent and send it back to the other room so that it can be reused over and over again. The waste, the actual waste, is moved from the system with a sludge pump in the bottom into this drum, in this particular case. Customers would directly feed it outside of the room. The first 2 evaporators are utilized in this case for the fractions that contain the compounds of interest. As the fraction comes out of the ICPC, it has about 90% solvent. The solvent level needs to be reduced, and this setup takes the solvent level down to about 25–30%. most of the accident, the methanol, comes out from the system. Then this process is finished with a set of rotary evaporators, because at that point it's mostly water and a bit of organic solvent together with the compounds of interest. These skids are used for solvent recovery from the fraction that include the compounds of interest. Very similar in operation to the purity solvent recovery solution. Certainly the settings are different in order to optimize for the compounds of interest and not to impact the material at all. Certainly the settings are different in order to optimize for the compounds of interest and not to impact the material at all. The solvent content in these streams are about 90% as they come out from the ICPC and will be reduced to about 25–30% by these machines. The final step is the rotary evaporation, where the solvent is taken out. At that point, the mixture mostly contains water. These newer version evaporator units have been increased in height for easier serviceability. These two evaporator sets are one version apart. The ones in the back are somewhat taller. That is due to the fact that the bottom of the vessels have been lifted a couple of inches for easier serviceability. The last step of the solvent recovery, following the COI evaporator, the material with the compounds of interest includes about 25–30% solvent. That solvent is mostly water and some hexane and methanol. In order to remove those from the oil, roto-evaporators are utilized most often. Hydolf, our strategic partner, developed custom solutions for recovering the solvent from these fractions. The customization covers a number of changes to Hydolf's standard industrial evaporator solution. It has been adapted to provide heat in the lines to prevent crystallization in the lines. The ember flask is also protecting the valuable material while the solvents are being recovered. These systems are highly automated. They have automatic vacuum control. That's a very important element, so no bumping happens. Therefore, any hazardous solvent staying within the system would not be emitted into the air, into the environment. These machines are to be placed in a non-hazardous zone. Usually, they are on the other side of the wall, where the COI evaporators will be placed. The systems have two options to handle the chilling needs. They can have dedicated chillers provided by Hydolf, or they can be configured to be serviced from a central main chiller. The problem, the potential challenges, with one chiller that the capacity should be the same for each of the Hydolf evaporators. That requires a parallel line, as opposed to sequential configuration. These are highly reliable machines which can be left alone, operating 24/7 overnight without human supervision. Thank you for joining us.
Welcome back. I hope you enjoyed the virtual tour of our CPC platform. So now I will take some time to answer questions from our viewers. So let's see, first up, how important is the auxiliary system and its stability on temperature and flow rates, etc., etc.? So what you've seen in that running system in this video, in that full-scale system, there's a well-defined auxiliary system for the heating, so for the hot water, for the buffer water, and for the cooling system as well, for the evaporator bodies, for the heat exchangers. And all of them are PLC controlled systems that are actually giving a proper flow rate and creating a proper heat and mass balance for the evaporator bodies, for the heat exchangers, and also for the buffer tanks. So in a lack of those systems, you're going to have significant variation and performance alteration mainly in the evaporators. And one more important story is that on the buffer tanks, where the sample is also stored, the diluted sample that's going to be injected in batch mode, that tank is also heated and circulated in order to avoid any kind of precipitation and settling in the system that later on might cause clogging. Next question. What is C1D2 and why is needed? Well, since we have volatile flammable liquids and in that closed confined space, and we have, although as it says also in the video that it's in a closed loop, you still have in the input and the output of the system is an open system where the customer feeds and then a customer retracts the final product. So from that perspective, there are openings in that system, and that's why you are going, and since you are using flammable liquids like hexane or methanol, etc., you need to going to be extremely careful and create that safe environment as you've seen on the video. So that's why you need that specific C1D2 designation. Why do you need rotary evaporators at the end? Well, that's wide film evaporators that we selected to use in our complete CPC and COI process. It's mainly dealing with the bulk and the large volume of the solvent and reducing significantly the volume of that diluted sample and the fractions and impurities. But at the end, wide film evaporators are actually good up to, let's say, 90% of the solvents to be recovered, and you still need to be able to manipulate and get the final product out of that system. Some of those products, some of those components are actually easy to be crystallized in room temperature. So that's why we need to keep them in a certain volume, in a solvate form. And once you have that final, let's say, concentrate out of that system, that's where you're going to use the rotary evaporators. You're using a limited number of rotary evaporators because you significantly reduce the volume of the organic and inorganic solvents in that system. So at the end, you are okay with just using a couple of rotary evaporators. And the other important reason why you need is basically you creating products where you have to fulfill some requirements on solvent residues, and these high dose evaporator units are actually excellent for obtaining that low ppm level of solvents. Somebody was asking also about the maintenance schedule and how extensive is that maintenance and maintaining the instrument. Well, it's always, we used to say that it's always related to the customer and the operational condition, what's occurring at the customer. In most of the cases, the customer for continuous batch operation understands that samples, solvents, etc., needs to be entered the system in a filtered and in a perfectly solubilized form. Once this is granted, then the system is running relatively smoothly. Maintenance-wise, there are regular maintenance schedules which can be conducted by the customer quarterly, monthly, daily. That is in our maintenance schedule written that is also discussed during the installation period. I guess that's what the list of the questions that we have time for now. So that concludes our Q&A session. If you didn't get your question, a member of our team will get back to you and with more details, of course. So additionally, if you have burning questions after this webinar, please send an email to sales@rotachrome.com. You can also access all of our previous webinars by visiting webinar.rotachrome.com. You can also reach us on Facebook, Instagram, LinkedIn, and Twitter. Thank you again for joining us today. We look forward to hearing from you, seeing you in a future webinar. Thank you.